Myself Dr. Deepika Saini from Lloyd Institute of Management and Technology. Today we are going to study about parasympathomimetic agents in the unit 3 of medicinal chemistry. The course code is BP402T. So, let us start with parasympathomimetic drugs, or we can say cholinomimetic drugs. As the name suggested, it is cholinomimetic, means that will mimic the action of acetylcholine. So, cholinomimetic, or we can say parasympathomimetic, as we have studied in the earlier lecture, that the neurotransmitter that is responsible for parasympathetic activity is acetylcholine. So, cholinomimetic or parasympathomimetic or the cholinergic drugs are the drugs that will produce a muscarinic action on the receptor of the effector organs by the postganglionic cholinergic nerves. So, basically cholinergic drugs are going to mimic the action of acetylcholine either on the muscarinic receptor or the nicotinic receptor, and will produce the action as produced by the acetylcholine. The only difference is they will be more potent, means they are going to exert this action to a greater extent as compared to acetylcholine. So, when we talk about cholinergic drug, the structure activity relationship is to be studied. This is the structure of acetylcholine. So, we have several options to make different type of substitution and to study what will happen if we will replace certain groups by different substituents. So, when we have seen this is the structure of acetylcholine and this is the general structure for cholinergic drugs. So, we need to mimic the action of acetylcholine. We have keep the basic structure as that of the acetylcholine, like the C double bond O, here is oxygen, here is oxygen, here is ethylene bridge. So, we have taken any alkyl group can be replaced and the nitrogen with two different substituents. So, now if we talk about structure activity relationship, we have different targets in case of cholinergic drugs. We can see the different substitution at this carbon, or we can change this steric group, or this the ethylene bridge in case of acetylcholine. This can be modified further, definitely the nitrogen atom. So, first of all we will start with the alpha carbon with respect to the ester group. So, this was the ester group and this is the alpha carbon. So, we have like maybe a hydrogen atom or a hydroxyl group, a hydroxymethyl group or carboxamide group. These all groups can be present in place of any of these substituents. So, the hydrogen atom can be an option when we talk about the hydroxyl group or hydroxymethyl group or carboxamide group. When hydroxyl group or hydroxymethyl group, they will generally the antagonist activity is usually more potent. On further substitution, it has been seen like they can produce even the antagonist property. In case of these R2 or R3, this can be any heterocyclic or carbocyclic ring. Carbocyclic ring like phenyl or cyclohexyl, cyclopentyl, and it can be any heterocyclic ring like nitrogen or sulfur containing carbocyclic groups. And for the maximum of the antagonist property, that will also lead to antagonist property. So, now if we have substitute the naphthalene rings, if we will substitute this R2 or R3 with naphthalene group at R2 or R3, that will lead to the formation of inactive compounds, because there is steric hindrance at the muscarinic receptor, and that will decrease the binding of the drug with the receptor. Hence, the use of the replacement of this R2 or R3 with the naphthalene ring is of no use, as it will produce inactive compounds. Now, if we have concluded, like if bigger R2 or R3 groups will bind to the hydrophobic region outside the acetylcholine receptor, and hence the main receptor will have least binding and will produce no effect corresponding to the acetylcholine. So, we have cleared this thing, like R1 is definitely with this hydrogen atom or hydroxyl group or hydroxymethyl group, but this hydroxy and hydroxymethyl will produce the antagonistic activity, and apart from that R2 or R3, they can be any carbocyclic or any heterocyclic compounds like phenyl, or we can say cyclohexyls. We can like have cyclohexyl or cyclopentyl group, anything in a carbocyclic form. But when we have substituted this R2, R3, if there is replacement with the naphthalene ring, that has lead to the inactive compound formation. So, these bigger groups will produce an hindrance at the muscarinic receptor, rather they will bind to the hydrophobic region, that is outside the acetylcholine receptor site. So, these drugs will not be able to reach the exact acetylcholine receptor site and will not be able to bind, and hence will produce no action corresponding to the acetylcholine receptor. And if we talk about hydroxy group that we have taken at R1, that will increase the binding strength, because that will participate in the hydrogen bonding, and as we know, their hydrogen bonds are quite strong bond when we talk about a drug and a receptor. So, this binding, this hydrogen bonding of the drug with the receptor, that will increase the binding affinity, the binding strength of the drug with the receptor. So, hydroxyl group will enhance the binding of drug with the receptor. So, the next category that we can opt for is alteration at the ester group. So, we can change this esteric group as well. So, ester group is as such not required when we talk about the cholinergic action, but the oxygen, the presence of oxygen atom is essential in this region to maintain the structure or to maintain the pharmacological action. So, another point is the ester group that is very susceptible to the hydrolysis, and that is the reason that acetylcholine is rapidly hydrolyzed through acetylcholinesterase. So, alternative groups like carbamates or the ethers or the ketones, they can be used to decrease this susceptibility towards the hydrolysis. So, that will lead to the compounds that will resist the hydrolysis. So, their metabolism can be delayed while the activity is whole soul maintained. So, when we talk about the ester group, it would be a better option to replace this esteric group through either through the carbamates or through the ether or through the ketonic functional group to reduce the chances of hydrolysis. Like the examples are, suppose if we have replaced this with the carbamates or with the ether, like here we have made it carbamates, either here the CH3 is being replaced. Now this is carbamate, NH2COO group is carbamate. So we have produced the carbamates. Apart from that, ethers, like we have taken the ether group, and or we can replace this COO moiety by the ketonic group, like the oxygen here is being removed to make it ketonic group. So these are the modification that can be made so that the hydrolysis can be reduced. The next is that the modification at the R4, this bridge position. So, as in the case of acetylcholine, there is ethylene group. So, there is a rule of 5. Rule of 5 states there should not be more than 5 atoms between the nitrogen and the terminal hydrogen. So, it means suppose if we have hydrogen here at this point and we have nitrogen here. So, the rule of 5 suggests the maximum of 5 atoms are allowed. 5 atoms, 1, 2, 3. So, the remaining possibility here is of 2 carbon to make this rule of 5. Like the ideally 2 carbon chain possess best activity. If we suppose that we should increase the chain length, but that will decrease the activity rapidly. So when we talk about this ethylene bridge in case of acetylcholine, hence the activity can be maintained if there is a 2 carbon chain only. Apart from that, if we try to introduce any kind of cyclic compound or to the or more carbon atom chain, that will definitely lead to decrease in the activity. Like addition of the methyl group in the ethylene, like if we talk about the methyl group addition in the ethylene carbon can affect the selectivity. Means if we will introduce either at the alpha or beta, but that will be defined by the position. Like suppose if we introduce an methyl group in this alpha position like this, hence but the one thing to note, the difference between the nitrogen and the terminal hydrogen, this one, the number of carbon atom should remain 5, 1, 2, 3, 4 and 5, that should remain 5. We are not introducing any methyl group in between this. We are substituting the ethylene bridge, this ethylene bridge at 2 different positions. Like suppose if we will include a methyl group at a alpha position, that with respect to the nitrogen, that will reduce the potency, but it has been seen the potency is, though the potency is being reduced, but still these compounds are selective to the nicotinic receptor. Similarly, if the methyl group is being included or introduced at the beta position, that will increase the selectivity towards muscarinic receptor. So, in this ethylene bridge, in this 2 carbon chain bridge, we have seen that if there is any addition of methyl group, that will definitely affect the selectivity of that compound. Like as we have studied earlier, like the methyl inclusion in the alpha carbon and alpha carbon with respect to the nitrogen present. Here is the nitrogen, so this is alpha carbon and this is the beta carbon. So with respect to the nitrogen atom, this alpha carbon that will reduces the potency, but the compound that will be formed through the inclusion of this methyl group at alpha position, they are quite selective for the nicotinic receptor. So, to make the drug selective towards the nicotinic receptor, the one thing to point out, one thing to note down is that there should be methyl group at the alpha carbon position. Further, when the methyl group, this is introduced at the beta carbon at the position relative to the nitrogen, that will, and apart from one more thing to note down, that it reduces the potency when the methyl group is introduced at the alpha carbon, but the activity is remain same. The potency has been maintained when the methyl is in when methyl inclusion is done at beta carbon of acetylcholine, and that will be more selective towards the muscarinic receptor. Like if we talk about, like in this case we have seen alpha position, this alpha position, methyl introduction, and in this case there is a beta, at beta position there is methyl substitution. When we talk about activity, it is similar, it is not active as that of the acetylcholine, it is similar as that of acetylcholine, it is not clinically used, but these are selective to the nicotinic receptor, these are being clinically used, and these are selective to the muscarinic receptors. The last one is we can do the substitution at the amine group, and this is one of the most important substitution to be considered. First of all, the presence of nitrogen at the quaternary ionic form means the nitrogen is being substituted through 4 positions and will contain a positive charge. This is quaternary ionic form. This is very important when we talk about the agonist activity. To make them agonist to the acetylcholine, the nitrogen must be in quaternary ionic form. Quaternary ammonium drugs are basically used in the treatment of ulcers or other conditions, and for which the reduction in gastric secretions and also reduce the motility of gastrointestinal tract are desired. So, there the quaternary ammonium drugs are being used. Further, if we replace this nitrogen atom with other elements like sulfur or arsenic or phosphorus, that always reduces the activity. Means the presence of nitrogen is mandatory in terms of activity. Like if we have like replaced this nitrogen with a sulfur. Now, after the replacement with the sulfur, that will decrease, that will here the positive charge on the nitrogen that was present previously is being removed after the addition of this sulfur atom, and hence that will reduce the activity. If we will introduce arsenic or phosphorus, that maintain that charge, but still the binding affinity is not that sufficient to produce the same action as that of acetylcholine, and hence the activity is again reduced when we have replaced any of the atoms with the nitrogen atom. Further, now the next thing is nitrogen is essential, and apart from that replacing the nitrogen atom with any other atom, that has reduced the activity. So, now the substitution of nitrogen atom is also being considered. So, first of all the substitution at the nitrogen atom. If we substitute with the methyl group, that will lead to the active compounds. When we introduce another atom like methyl is being replaced with the ethyl group, the activity suddenly switches from agonist to antagonist. Now, the ethyl containing nitrogen atom compounds are being those compounds will be those developed compounds will be antagonist in nature. Further increase in carbon atom chain like propyl or the higher alkyl atom, the activity is completely abolished. So, they will not possess any kind of activity. If any of the R is either ethyl or propyl, the compounds are found to be active, but will be less potent as compared to acetylcholine. If any alkyl group is replaced with as we have seen, there is if there is less number of carbon atom, activity is higher. So, if we suppose the alkyl group is being replaced with hydrogen atom, again the activity is reduced. So, when we talk about the substitution at the nitrogen atom, there are several options we have, like we have already told the quaternary ammonium, the quaternary nitrogen atom is required. So, we have various options, like if we talk about from here, it is as binded with this ethylene chain, and we have three options R1, R2 and R3. If these are substituted with methyl, that is okay, the compound formed will be active. If we have replaced this methyl group with the ethyl, the activity will be switches to the antagonist rather than the agonist to the acetylcholine. Further, if further there is any change, like if we increase the carbon atom and we will replace this methyl group with the propyl or the higher alkyl, the activity is completely abolished. And if any of the, so we have three options R1, R2 and R3. If any of the R is ethyl or propyl, the compounds are found to be active, but not much potent as compared to the acetylcholine. Or if any alkyl group is replaced with the single hydrogen atom, again the activity is being reduced. So, from this we have few points to note down. The first thing is, when we talk about the first methyl atom, so we should have a carbon chain, the methyl atom at the first position. When we talk about steric form, we can change this steric form to ether or to the carbamates or to the ketonic groups, so that the activity can be maintained by reducing the chances of hydrolysis. Then further, through the rule of 5, we have studied that there should be at least there should be not more than 2 carbon chain between the nitrogen and the steric group, and that afterwards we have seen if we replace nitrogen with any other atoms, the activity is being decreased. So, after this, further, the last thing to note down, when we talk about different substitution, only the methyl atom will produce the agonist activity after the addition of ethyl group or introduction of ethyl propyl. These all lead to the either to the antagonistic activity or the abolishment of the activity. So, this is all about the structure activity relationship of parasympathomimetic drugs. Thank you very much.